Okay, here goes. So I'm going to teach you photosynthesis using my patio table and a bunch of Legos. So let me give you an overview here. Right now you're seeing the outline of the chloroplast. So this is the outside membrane of the chloroplast. And then these are the thylakoid discs. So a bunch of those would make up what's called a granum. And what we're going to be zooming in on is a process called photophosphorylation, which is the first part of photosynthesis. And then we're going to move on to the Calvin cycle, which you can see way on the left, which happens in the liquid part of the chloroplast called the stroma. So here you can, I'm zooming out, you can see everything. Okay, and I'm going to zoom in. Hopefully the wind's not going to blow everything away. Okay, so photophosphorylation. So as the name implies, it's going to use light to phosphorylate phosphorylate ADP to make ATP. So here this is going to seem really similar to oxidative phosphorylation because you're going to get a proton gradient, and you have our friend here, ATP synthase, which is going to use the proton gradient. We have our friends ADP and P, which will go together to make ATP when the protons are flowing through ATP synthase. Okay, so here we go. So what's going to start this whole process is light. So here, this is called a photosystem. And the photosystem is an integral membrane protein with all kinds of stuff. It's got a bunch of chlorophyll molecules, which I'm showing you here. And there's also accessory pigments, which I'm showing you in yellow and red. These are other pigments that help to gather more wavelengths of light. So chlorophyll is a molecule embedded into the phospholipid bilayer of the thylakoid space. And chlorophyll is green because it absorbs blue and red light and reflects green light. So when light hits this photosystem, the chlorophyll absorbs that light energy, and this is called the reaction center chlorophyll, and it gets so much energy, relayed in from all these pigments, and the electrons that are on the ring of the chlorophyll start to just go crazy, and the outermost electron just leaves the orbital and is going to jump up a level and be accepted by this first electron carrier here in the membrane. So we're going to get an electron transport chain just like we did in oxidative phosphorylation. So you'll have electron carriers, one more consecutively electronegative than the other. But now this chlorophyll is missing an electron, and that's not a happy situation. So water here is going to actually split apart, and the electrons are going to be ripped out of the bond here between the hydrogen and the oxygen. And so what will happen is this water splits, and the electron will go here to replace the electron that was lost when the chlorophyll lost its electron. Meanwhile, the proton is going to get pumped as the electron moves down the electron transport chain, beepity beepity. It's going to get pumped into the thylakoid space. Now notice, in oxidative phosphorylation, we had a proton gradient between the inner membrane and the outer membrane, so it was sort of outside, like it got pumped out. And here the proton gradient is forming in, so here the pH is going to go down inside the thylakoid space. Okay, so let's go back to where the electron is. So there's another photosystem. What's weird is this is called photosystem 2 and this is called photosystem 1, which I could explain why, but anyway, this in sequential order it doesn't really make sense. So here photosystem 1 also is is absorbing light energy, and this electron is going to jump out of its orbital and be accepted by this electron carrier. And so now it's missing an electron, but it gets replaced from photosystem 2. So continually, we're going to keep continuing to do this process. Eventually, what will form as we move electrons down the chain, eventually when this all happens, is we'll get oxygen gas. So oxygen is a byproduct of photosynthesis from the water splitting and losing its hydrogen. And this oxygen is going to leave the plant, and of course that's the oxygen that we use as heterotrophs. But also don't forget that plants have mitochondria as well, so that the mitochondria of the plant could use oxygen as well. Okay, so meanwhile, back here at the last electron carrier, the final electron acceptor, let me make sure that you can see this. I'm going to scoot this over a little bit so you can see over here better. The final electron acceptor is NADP, which is an electron carrier just like NAD was. It has a phosphate group. So this is going to pick up the electrons, and it's going to pick up some protons as well to become NADPH. Okay, so that's NADPH. NADPH is going to be used in the Calvin cycle. So this is going to be the hydrogen source to make sugar. So that's going to go into the Calvin cycle later. Okay, meanwhile, you have this proton gradient, which just like before is going to go through the little pore here on the side. It's going to cause ATP synthase to spin, and then the phosphate and the ADP will go into the active site of ATP synthase, and when it spins, you'll be able to phosphorylate the phosphate on ADP and make ATP. And so this ATP will also be used in the next part called the Calvin cycle. So notice that you have other photosystems on other thylakoids. Of course, each thylakoid has more than one of these systems that's going to produce the proton gradient. So the products of photophosphorylation are NADPH and ATP. And both of those are going to be used in the Calvin cycle to make sugar. So now I'm going to switch that video and we're going to end this video.